Welcome to Lecture 13, Mutation. In order to think about how mutation affects genes and affects proteins and affects coding sequences, it's important to remember the flow of information within the cell. DNA, all right, the nucleic acid, encodes for genes, which are transcribed into mRNA. All right, and so there's a linear stretch of sequence here. ATG would be indicating the start codon, and then codons in sets of 3, which get transcribed into mRNA. And you can see that the T is replaced with a U or a uracil right there, and again in sets of 3. And after transcription, translation occurs, in which the world of nucleic acids is left and the world of amino acid sequences is entered. And in this particular case, we have a stretch of methionine, serine, threonine, so on and so forth, which creates what is called a polypeptide or an amino acid sequence. So transcription, because we're staying in the world of nucleic acids, is basically copying the nucleic acid information from a DNA strand to an mRNA strand. But when you change the language, when you go from nucleic acids to proteins, that's what we call translation. Now, just to remind you, there are some critical differences between RNA and DNA. Shown right here are the ribose sugars, reminding you that at the 2' carbon in DNA, or deoxyribose nucleic acids, the sugar part, the ribose part, the 2' hydroxyl, the 2' carbon does not have a hydroxyl group at that position, but in RNA, the ribose is not a deoxyribose, it's just straight up ribose, and at the 3' and the 2', so 3' here and then 2' right here, there is a hydroxyl group. And another critical difference between RNA and DNA is that in DNA, the base is thymine, but that base that is thymine shown right here, in RNA, the methyl group on the thymine base is replaced with just a hydrogen, and then therefore in RNA that base becomes a uracil. So aside from those two differences, number one, the 2' hydroxyl instead of the 2' hydrogen being present in DNA versus RNA over here, and the uracil instead of the thymine. The growing end of the 3', the growing end of the RNA is likewise occurring at the 3' end. All right. So in this particular case, we have transcription, which a DNA strand here. Here's the 5' end, and here's the 3' end. Again, in an anti-parallel fashion, where the 5' end is opposite the 3' end. Here, now we're extending this particular molecule, growing an RNA chain. The 3' end of the RNA is extending. This next nucleotide in line is matching up with this A here, this uracil, and then you have the 3' end growing. But what you can remember is that while it's a 3' end of the RNA that's growing, the template of DNA shown here in blue is read from the 3' to 5' direction. All right, so the 3' end grows, but the template itself is being read in that direction that way. Now, proteins are the products of RNA translation, and oftentimes we show proteins as these large sort of like blobby features or figures like this. But keep in mind that amino acid sequences or polypeptides are also linear sequences, similar to DNA sequences or RNA sequences. But RNA and DNA don't have these interesting properties, or to this extent that proteins do, in which the 20 different amino acids have interesting features that allow them to fold up and form these sort of large, sort of blobby structures. Okay, so here's a representation of a protein sequence. Here's a representation of another protein structure. But keep in mind that inside this sort of blobby structure is a single long amino acid chain that folds up. And it's the way that proteins fold that determine their function. So some proteins you might know, these are structural proteins. So for example, keratin, hair and nails, or actin, which forms the structural component of the spindle that in mitosis that pulls the chromosomes apart. There are metabolic enzymes. We've talked a little bit about enzymes that contribute to metabolism. One might be alcohol dehydrogenase, which metabolizes ethanol. Of course, there's also transport that, such for hemoglobin, moves oxygen molecules around in your blood. So to think about this more in greater detail, because the subject of this lecture is mutation, it's important to remember how the code of nucleic acids is translated into RNA. And so in this particular codon table, we represent the standard genetic code. Not all organisms obey this particular table here. But what we're showing is the three positions with respect to the nucleotides, and we're showing them in RNA. We're not showing them in DNA because it's the RNA that gets translated, not the DNA directly. And in this first row here, and then the second row, and then third row and fourth row, we have the corresponding positions for U, C, A, and G in the RNA. All right, and then this column, this column, this column, and this column is the position for the second nucleotide. So U there, all right, for the second position, or C there for the second position. And then you also would have an A for the second position, or a G for the second position, and then the row within these sort of larger rows is the third position. So in this particular case, U, C, A, G. All right. So we'll walk through how this particular codon table influences the ways that mutations change the function of proteins. So it's important to remember when you're looking at this that there are some very important codons that determine the beginning and ending of translation. So an mRNA molecule, the first AUG oftentimes, and this is not always, but the general rule is that the first one would be initiating translation and also initiating the methionine addition to the growing amino acid polypeptide sequence. So AUG codes for methionine, it codes for often known as START. And then we have three codons which typically represent the stop codons, UAA, UAG, and UGA, and they terminate, they stop the translation of the mRNA molecule. But keep in mind, termination or stop, those are not names of amino acids. So at that position, an amino acid is not actually put in. So let's look at this table a little bit more closely with respect to how different types of mutations within that framework of the three base pairs of forming a codon, how the different types of mutations can affect the sequence of amino acids. So what we're showing here is the DNA sequence, right? And you know that that's DNA because there's a T there instead of a U. We're showing the consequences of the DNA sequence mutations. Now, at the molecular level, arginine, let's just go back here, arginine can be encoded for by, say, for example, AGA or AGG. And so let's suppose that this original codon was AGA encoded for an arginine. Well, you can see here there's some redundancy. AGA and AGG both code for an arginine. So a G mutation that occurs at that particular position would also still encode an arginine. So a mutation in the DNA sequence in this way does not change the underlying amino acid sequence. And this is what we call a synonymous mutation. So the altered codon specifies the same amino acid. However, in this particular case, we have a mutation. Instead of at the third position, we have it at the second position. We change this codon A, G, might be G, for example, to AAA. Here, this mutation actually changes the amino acid sequence. Likewise, this ATA changes the amino acid sequence. You can go back to the codon table and see that AAA, let me see if I can find it, right here is lysine, right? There is lysine. Now, both of these changes in this second position, all right, from AGA to AAA or AGA to ATA, both change the amino acid sequence, and therefore they're called missense mutations. But the different amino acids have different chemical properties And so in this particular case, arginine is somewhat similar to lysine, so we would call that a conservative change because the altered codon specifies a chemically similar amino acid. However, if there's a drastic change, for example in the case of isoleucine, the altered codon might specify a very dissimilar amino acid, and in this particular case we would call this a missense mutation that is non-conservative. And then finally you can have sequences changes that occur, and here the sequence AAG, a mutation has been induced that causes a TAG codon to form, and then because of that, that causes a stop, and that will be called a nonsense mutation, and that would prevent the further translation of any amino acids downstream of that particular stop codon mutation. So here you can see that within the codon table you have UCU, UCC, UCA, UCG. Here in this case the third position, all changes to this codon within this sort of framework, within this box at the third position don't change the amino acid sequence. So they're always serine in this case. So this would mean that at this particular codon all third position changes are quote silent or synonymous. However, in this particular case, let's look at the first position. If the first position of the codon, which is a nucleotide, if there's a mutation that changes to the G nucleotide, well, that would be causing a missense or non-synonymous mutation because all A's converting to G's would convert this codon from a threonine to an alanine. Now, non-conservative missense mutations can cause all sorts of different diseases. For example, sickle cell anemia. Here's a particular mRNA sequence that has a codon sequence CCU, GAG, GAG, AAG. And in this particular case, this is the wild type structure. This triplet set of codons would be translated with pro, glu, and lysine, and glutamic acid would be at this particular position. However, in this particular case, a mutation can occur. Instead of GAG, the codon would be GUG, and then this would change the glutamic acid to a valine at this particular position. So what this does is this changes the structure of hemoglobin inside the red blood cells. So here are normal red blood cells, and they flow smoothly through the capillaries and blood vessels, and inside there would be normal hemoglobin. But in the case where this non-conservative missense mutation occurs inside hemoglobin, it causes the hemoglobin to fold improperly, and then that has consequences for the structure of the red blood cells. The red blood cells form this sort of sickle shape, and they can get stuck inside the capillaries or the blood vessels. So this is a case of a non-conservative missense mutation that happens when this second base pair within this codon is mutated. And then reminding you that the way you would get a nonsense mutation, not all codons have the ability to mutate to a nonsense stop codon, but in the case of tyrosine, if this U or C is mutated into an A or a G, we would convert a triplet codon from tyrosine to a stop codon. Now, in addition to what we call point mutations, or basically like single mutations that happen and change a base from one to another, you can also have insertions or deletions, and that can have very profound consequences. For example, this particular sequence A, C, G, T, G, A, G, G, A, G, 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 if the G is deleted from it, then in that particular case, what that can do is it can change the entire reading frame, because the reading frame depends on a sort of ordered series of 3 and 3 and 3 base pairs, one following after the other in a particular codon-based sequence. So here we have an insertion, and what that does is normally that codon that would be right after this threonine would be AAG, but a G gets put into it, and so instead of the original sequence being AAG determined or AGA determined, now it's GAA, GAG, so on and so forth. And what that does is it changes all the amino acids that are downstream of that insertion. Likewise, a deletion can also cause a frameshift mutation because here a 1 base pair deletion has been excised from the DNA sequence. The A here is gone, and so then the translation actually occurs in the form of AGA, GAG, and that will change also the downstream nucleotide, the downstream amino acids. So which ones of these will cause a frameshift mutation? A 2 base pair insertion will, because it will shift the frame off by 2 base pairs, and the frame is in 3 base pairs. But a 3 base pair insertion won't, because it's in multiple of 3, right? So it will cause the insertion of an amino acid, but it won't cause the entire frame to be messed up. An 8 base pair deletion would cause a frameshift mutation, again, because it's not a multiple of 3, but a 27 base pair deletion would not. The 27 base pair deletion would lead to 9 amino acids deleted, but the frame would be maintained downstream of that deletion. Now, if you look at codons, you could see, if you stare at the codon table for long enough, there are some places where any mutation does not actually change the amino acid sequence. So we showed a case with serine, for example, that mutations at the 3rd position didn't change the amino acid sequence. We would call those 4-fold degenerate sites because all 4 nucleotides can be interchanged with one another and the amino acid sequence would be the same. Now, 4-fold degenerate sites have that property. Non-degenerate sites, and those are oftentimes 2nd position or 1st position. Non-degenerate sites is if you change the nucleotide at all, what that does is it will change the amino acid sequence. And 2-fold degenerate sites are sort of in between. If you have, one nucleotide mutation might not change it, but 2 of the other 2 mutations might. So, for example, if you have an A nucleotide at a particular position, a G nucleotide mutation might not change anything, but a T or a C might. And so those would be called 2-fold degenerate sites. Now, what's interesting about those is that if you actually take coding sequences, say, for example, hemoglobin, and compare them across species, you can line those sequences up and you can see where are the places where changes are accumulating the most. It turns out, and this is in terms of units of substitutions per nucleotide site per 1 billion years. This is a very large time scale, but you can make these inferences based on how much time you think is diverged between, say, for example, humans and chimps. You can look at the substitutions and you can see that the rate of accumulation of substitutions is much higher, say, for example, between us and chimps or us and mice at 4-fold degenerate sites compared to 2-fold degenerate sites. And 2-fold degenerate sites are going to have an accumulation of differences that is happening at a faster rate compared to non-degenerate sites. And why this is because the changes between us and chimps or us and humans that change the amino acid sequence, like for example these ones, are going to be more drastic and more likely to be harmful and more likely to be eliminated in the evolution of divergence between 2 species. However, nucleotide changes that don't change the amino acid sequence are more tolerated in the divergence between 2 species. So you'll see a greater accumulation of changes here. And what this means is if you were to compare codons between us and chimp, we would see that the nucleotide differences, usually at the 3rd positions, would be greater than the abundance of the nucleotide differences at, say, for example, the 1st or 2nd positions. Now, where does mutation come from? It comes from it comes primarily from replication. It can happen, so in this particular case, DNA sequence is being generated, and this is the template here. And so, the A with a T, the G with the C, the G with the C, the DNA polymerase here has made some sort of mistake. And instead of putting down a T, a G is mismatched there. Now, oftentimes that mismatch is recognized by the DNA polymerase as proofreading, but not always. So you can see that a mistake by DNA polymerase can introduce a mutation. The other thing that can also happen is you can introduce insertions, or in this particular case, deletions, because you might have sequences that are repetitive, like CT, 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 CT. And in this particular case, there's like these repetitive sequences can loop out. And so in this particular case, if a loop out occurred, then the DNA that's being replicated off the other strand would just skip over that CT. All right. So in the next round of replication, this CT is missing on this duplex, but is still present on this duplex, so you can get deletions that way. And for similar reasons, you can also get insertions because of basically stuttering on the part of a DNA polymerase in a sequence that's repetitive. So how those replication mistakes are perpetuated is kind of shown in this diagram here. Here you might have a nucleotide sequence that normal, not mutant, for example, and then DNA replication occurs, and then one of the strands gets replicated up here, and then the other strand gets replicated down here, and then this is the template, and then the nucleotide that gets placed at this position is wrong. All right. Now, DNA polymerase usually will identify that and fix that mistake, but that's not always going to be the case. And if that mismatch remains, all right, that mismatch remains through one more round of DNA replication, well, this A will get rematched back with a T. But the G here now instead will get matched with the C, and now you have both nucleotides fully matched, but that's an error that was propagated from this prior round of DNA replication, and that error then will be passed on to all descendant DNA strands throughout cell division, or throughout maybe off to the next generation if it gets transmitted in the form of a gamete. And then again, replication slippage, which I said before, can be a powerful form of mutation. And we've talked a fair bit about Huntington's disease. This is a person who has Huntington's disease, and it's, I'll remind you, is a neurodegenerative disorder that can cause this type of problem with the hands, and it can cause tremors. And it again arises from this sort of stuttering that can occur inside DNA sequences. The Huntington disease mutant alleles have an unstable number of CAG repeats. So CAG, 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 which encodes for glutamine. And this repeated sequence, if it folds back on itself in this particular way, you can actually have sort of like the DNA polymerase can stutter. This sequence here is extending. It folds back here, and then it re-extends and amplifies the number of repeats. And this has a very interesting property in that it's, quote, accelerating within pedigrees, because there may be, maybe 60 or 70 copies in a normal individual, and then there might be like 90 or 100 copies of it in an individual that's somewhat affected by this disease and maybe have a late onset Huntington's. But because it amplifies, it can then amplify to a greater extent each generation. And so across generations, you'll get further and further amplification of this unstable sequence. And people that have larger numbers of repeats of this sequence because of this sort of replicative process will have Huntington's disease symptoms start to arise at an earlier time in their life. So, in a pedigree, you might have individuals that show signs of Huntington's disease at an older age, but then in later generations, you might have Huntington's disease showing up in individuals that have younger ages. So that's DNA replication, but DNA damage and DNA mutation can also actually be induced directly from chemicals that you generate within your own body, basically induced by metabolism through oxidative damage. A potentially harmful byproduct of metabolism are oxygen radicals, here in the format of a hydrogen peroxide molecule, and those oxygen radicals can damage bases. In this particular case of guanine, this hydrogen here has been oxidized and an oxygen has been placed there. Now that changes the base from a guanine to something known as an 8-oxoguanine. Now if it gets repaired, it's okay, but if the 8-oxoguanine is maintained, so here we have a G and a C, which is a normal base pairing, and then we have a oxygen radical change that G to an 8-oxoguanine. In the next round of DNA replication, if it's not repaired, an A will be actually matched to that instead. So the DNA polymerase has a preference for putting an A against an 8-oxoguanine instead of a C. Now, if another round of DNA replication occurs, then the A will be retained and then a T will be then put in its place, and that would then lead to a mutation that becomes fixed in the descendant lineage. So here, mutation is caused by metabolism, basically chemicals being produced in your own body. But then there are also mutagens, which are chemicals that come from outside of the body. And so the most famous one, I would argue, would be ethyl methanosulfonate, or EMS here, all right, or nitrogen mustard. These compounds are known as alkylating agents, and they will put alkyl groups onto bases, and that will, again, disrupt the base pairing. And so EMS, this one right here, EMS, is actually probably one of the more commonly used mutagens in laboratories. I told you at the beginning of the class that oftentimes geneticists would use X-rays or UV, but now most mutation that's induced randomly is induced by this EMS compound. And so in this particular case, this EMS compound converts guanine into O6-ethylguanine. And that again changes the base pairing properties of guanine in this particular case too. Instead of matching with G with C, instead the O6-ethylguanine basically prefers base pairing with thymine. And so then once the GC base is converted to this sort of O6-ethylguanine thymine, then another round of DNA replication will fix an AT base. And then another form of harmful mutation is UV irradiation. This has a particular consequence with respect to thymines, that UV irradiation can generate crosslinks between thymines that are right next to each other in a helix, which form something called thymine dimers. So what this does is the presence of these UV radiation events that cause these thymine dimers, this causes the base pairing of T with A to be disrupted, and then DNA polymerase has a hard time of correctly putting the A at the right base during DNA replication. And then we've also talked about X-rays, another type of important mutagen. And they can cause nucleotide damage, so they can cause point mutations like we talked about. But they can also break the back of DNA itself. They can break the strand either in the form of a single strand break, or in fact, most dangerous, they can cause a double stranded break. Now in this particular case, you have maybe an X-ray that directly ionizes this particular atom, and an electron flies off and causes damage. But the ionizing effects of X-rays don't just have to be through direct effects, but the ionization can actually lead to the formation of these oxygen radicals here. So this H2O, one of the electrons is knocked off, and then the oxygen radical is formed through the breakdown products of that particular, and it's the oxygen radicals that can then cause damage. Now, to understand how certain compounds are mutagenic, this is something that the FDA, the Food and Drug Administration, clearly wants to be able to do to make sure, for example, that the sodium benzoate or whatever in your Red Bull is not mutagenic. There has to be a way to basically test whether compounds are going to cause mutation. Now, one way you might think that you could do this to test if compounds cause mutation is you could say, well, I'm going to take the compound and I'm going to mix it up with a bunch of bacteria, and I'm going to see if I induce a bunch of mutations in the bacteria. But there's a problem with that. And the problem is that compounds that are not directly mutagenic can be processed into mutagenic compounds by metabolism. So you might take your sodium benzoate or whatever from the Red Bull, and you might put it on some bacteria, and